Hello and welcome everybody to the transplant video tutorials about resources for the Barley genome. My name is Manuel Spanagel from the Helmholtz Center in Munich. So if you want to find out more about the transplant project, please just visit our website here. And here is a brief overview about the tutorial so that you can jump to a specific chapter that you are interested in. So now a few words about the Barley genome. The Barley genome is very large, very complex, and the resulting genome resources are heterogeneous as a result. So this tutorial should help you to navigate through the different resources that are available and help you find things there. First part is about to find resources for the Barley genome. For that, you can access the Transplant Genome Resources Registry, which is available from the transplant website or the MIPS PLANTs.db webpage. You can search the registry by a keyword, and from the results you can directly access the corresponding webpage or resource. Each entry has its own report where you can access additional information such as the last update, the URL or the version number. Now if you are looking for a BLAST server for Barley, you come across this Barley ViroBlast instance hosted by IPK, where you have the keywords BLAST and the URL, and a detailed explanation of the fields is just below on the page. For the next part we are making use of the BLAST server provided by IPK. Here you can search your sequence of interest against the Barley whole genome sequence assembly as well as the genes. Okay, so here you can choose between the different data sets, for example the assembly or the Barley high confidence or low confidence genes. And here you can choose the BLAST version, for example BLAST N for BLAST of nucleotide sequences, or BLAST P for BLAST of protein sequences. And now as an example we are pasting our sequence here in the field. And we are choosing the correct BLAST version, so BLAST N for nucleotide BLAST, and the corresponding sequence set that we want to BLAST and search again. And then we click basic search. Now we are blasting against the high confidence genes of Barley, and you can also select some of the options of BLAST. Now we are waiting for the BLAST job to be processed. So the above panel gives you a visualization about the BLAST results that we have here. In red we have a very good hit against the Barley gene, and the lower panel gives you more information about that gene, like the identifier, the score, the identities, percentage and expected e-value. And the panel below then gives you more information about the alignment of that hit. So you can see this is a very good alignment here. And now we are interested in more details about the Barley gene that we identified with our BLAST search now, of course, and for that we are selecting this mlog Barley gene identifier here. And in the next tutorial we are going to retrieve some more details about that gene, and for that we can use the MIPS PLANTs.db or Ensemble Plants webpage. Both databases are hosting the Barley gene annotation, so we copy our gene identifier from Barley and we are visiting the Plants.db page from PGSB, formerly MIPS. So we are navigating to the Barley genome instance here, where you have a lot of information about the genome annotation, the genome zipper and so on. So for now we are visiting gene annotation because we are interested in the gene. As you can see, you got a search interface here where you can paste in your gene identifier from Barley, and you click search. And then you will see the search results here. So we find the gene identifier, and here's the element report where you have a lot of information about that particular gene model, such as the element type. You got a human readable description, you got a comment, you got the contig where the gene is located on, and you got information about the sequences that you can click and download in faster format, for example. You also got the protein sequence here. And there is some information about the gene family. For example, you can identify orthologous genes from other species from that. Here's some information about the author-MCL run that identified the orthologous genes, and here are the orthologs in a number of species, including Areopsis, Brachypodium, maize, and rice. And here's a number of links to internal or external resources that are related to that gene model. For example, the EnsemblePLANT's Barley instance. EnsemblePLANT is also hosting the Barley gene annotation, and that will be the next part of this tutorial. So here we are on the summary page of that particular Barley gene model. As you can see, there is a number of splice variants annotated. They are marked with 0.1, 0.2 or 0.3, and you can see that very nicely in the genome browser. So there is some information about the transcript available. If you click the transcript here, the particular 0.1 splice variant, you get some more information about that one. So for example, you can show the transcript table where you have a summary of the splice variants. So if we go back to the gene summary here, and on the left hand there is the navigation panel from EnsemblePLANT, where you get a lot of information about the particular gene model. For example, the GO, gene ontology terms for that gene. Here are the GOs from biological process. Here are the ones from molecular function category. So next we are going to have a look at the gene tree within Ensemble. This is a very useful tool to explore the phylogeny of the particular gene that we are interested in. So if you go there, you will see not only the orthologs but also the paralogs within most of the species that are included in EnsemblePLANT for your particular gene, and that is a very nice overview here. So you can see genes from, for example, Brachypodium, from Triticum, and so on. So here's the view about the orthologs that were identified for the gene. Gives you a very nice tabular overview about these ones. It also includes links to the corresponding gene summary report. It includes the percent of identity to the query. And as we go back to the summary here, we will learn some more about the Ensemble genome browser. So that's a representation of our gene model, including the splice variants. And if you click on region in detail, you get a more refined view of that region on the chromosome where your gene is at. As you can see, now you also have a display of the neighboring genes of the corresponding complete region on the chromosome. And now we can customize our view in that we can select active tracks that we want to see, our favorite tracks. For example, we can display more tracks related to sequence and assembly or to the synteny. Here we are selecting the track to display synteny with Brachypodium tachyon, which is a close relative. And as you can see, the track is then added onto the genome browser. So, in the next part of the tutorial we are also talking about synteny. Synteny is the conserved gene order between related species, and we are to investigate the syntenic relationships between cross-reference species such as Brachypodium and rice. And for that we are visiting the PLANTSDB Crow's Nest tool, and you can find links to that one in every gene report in PLANTSDB. So for example, here in the element report of the Barley gene that we had before, you can directly click to the link and you are directed to the Crow's Nest 2. Crow's Nest is a synteny viewer, and there are different levels of hierarchy for the visualization of the syntenic relationships between species. So it goes from level 1 up to level 4, where level 4 is the most detailed view and level 1 is the most general view. So what you see here is a microsynteny view between rice chromosome 5 and Barley chromosome 1. And all these lines represent syntenic relationships between orthologous genes. And as you can see, if you mouse over these relationships, you will always see the gene identifier that is responsible for that. And you can even jump from these links to the corresponding PLANT-DB element report, where you get more information about a particular gene. So if you go back here, you can also jump around the chromosome and move to a different position. And if you want to change the hierarchy level, you just choose this bar over there, where you can, for example, now go to level 2, and that is a representation of Barley chromosome 1 versus rice chromosome 5 and chromosome 10, where you can see syntenic relationships between that Barley chromosome to 2 different rice chromosomes. And again, if you mouse over these little lines, you will see the gene identifiers that are responsible for that pairing. And by clicking into the picture, you will zoom in to the corresponding region. And if you reach a certain level of detail, you will automatically be directed to the level 4, which is the most detailed level again, where you have an overview in the top picture and a more detailed view about the gene models that are giving the syntenic relationships between that pair. And again, you have the gene models here by clicking over. So another representation of level 2 is given in that view here. So this is a circular view of the syntenic relationships between the rice chromosome 5 and all the other rice chromosomes, as well as Barley chromosome 1. And level 1 is the most general overview, so you have all the chromosomes of Barley here, together with all the chromosomes of rice represented here. Another way of representation of the syntenic relationships between Barley and rice is given in the next view. So that one here. Here we see syntenic relationships between chromosome 6 in Barley and chromosome 2 in rice. And now I'm going to show you one more way to access the Crow's Nest 2 without going to the gene element report. So you can also access the Crow's Nest 2 directly from the PLANT-DB homepage. So there's a link to Crow's Nest under projects here. And if you want to enter Crow's Nest, you just need to click Enter Crow's Nest, where you will find a navigation bar where you can choose a target genome, for example Barley, and a reference genome, for example rice or Brachypodium, and you can then specify your target chromosome or your reference chromosome if you want. And by clicking Submit, you will get again the view from level 3. And if you click in, you will zoom into the view. And you are again in level 4, where you have the overview of that particular region and the more detailed view about the gene elements. Okay, and in the next part of the tutorial we will show you about how to retrieve and investigate a gene expression profile for a selected Barley gene. For that we are using the PGSP expression browser, and again you can access that too from the gene report of the particular gene that we had before by clicking here, and you will end up in a view of the gene expression browser. but there is also a number of additional entry points to the expression browser, such as by keyword or by gene. So this is the input page for the expression browser, and as you can see, you have search options for keywords, for gene identifiers. There is an interface for blast search, which we will show you next. So that is, you can put in your sequence of interest here. And there is the option to search by a gene list if you have more than one gene identifier that you want to search for. And going back to the keyword search, you can, for example, search also by GO category or by interpro ID. So now we are pasting in our Barley gene that we had all the time before. But before we search for our gene, we want to learn more about the RNA-seq experiment and the conditions. And you find that information on the lower part of the page. And if you want to learn more about the expression browser, there is a publication which you can review here. So now we are going to click submit, and with that we are accessing the overview page about the expression strength of the particular Barley gene that we are looking in different conditions. And if you click in here, we get to another representation of that, which is the expression strength in bars across the different conditions. And below we can access additional information, such as the expression in a tabular form, as well as some external annotation, and that includes the interpro IDs that were annotated for that particular gene, as well as the GO terms that were found here. There is a comment box where you can submit your comments. You will find the sequence, both nucleotide and amino acid, and you can also access the PLANTs-DB element report again by that link here. And you can also download all the expression data by using the download link on the left. Okay, so in the next part of the tutorial we are going to explore the Barley variation data for a selected gene model within the PLANTs ensemble instance. And for that we are visiting ensemble PLANTs, the Barley instance. We are entering our gene model that we always had before. We search for it, and we end up at the search results for that one here. So we are clicking the gene ID that we found, and we are accessing the gene report in ensemble PLANTs. We had that before already. We are clicking region in detail, and we are ending up at a more detailed view in ensemble PLANTs again. And once again we are using the left bar navigation panel, where we find genetic variation and resequencing, and we are clicking at the resequencing link here, and we are ending up at a representation report in ensemble PLANTs of the Barley genetic variation data. And as you can see here, there is a number of Barley varieties annotated and aligned. The different types of variations are given here, for example intronic, missense or synonymous substitutions, and the color coding highlights different types of variation at a position. Ensemble PLANTs also provides another way of representation of variation data, and that is directly within the ensemble genome browser. So to display the variation data, you will have to configure the track, and you will find a variation label here. So now we are selecting sequence variants. We want to have it as a density plot for now. We are selecting both, and we confirm. And now you will see the variation data for Barley in a separate track here in ensemble PLANTs genome browser. And if you zoom in a little bit, you will see the color coding for the different types of variation. such as missense variants, synonymous variants, and so on. And by clicking on the color box, you will get more information about the variation, such as the allele or the consequence of the SNP. By clicking that link, you will get more information about that SNP, for example, the location and the study, as well as some information about the genomic context and the genes in regulation. So by clicking at genes in regulation, you will get a nice overview about the consequences of the different variations within the gene of interest, such as a synonymous variant or a 5' UTR variant, as well as the allele and the position in the CDS. And you can get some more details about that by clicking at Show. So if you go back and you go to genomic context, you will get a nice graphical representation of the neighborhood of that variant. And again, by clicking at the color boxes, you will get more information about a particular variation. So in the next part of the tutorial, we want to use the Barley genome zipper to identify a putative location of a gene on the Barley genome. And for that, we are accessing the genome zipper at the PLANT.db web page. So we are going to the genome zipper page. We'll get some information about the genome zipper concept, and you get different menu points like data overview, search, or download. So if you go to data overview, we get a representation of the Barley chromosomes, and by clicking in you get a tabular overview of the genome zipper data at that position. So if you go back and we select search, you can search for genetic elements such as a gene, a marker, a EST, cDNA, and so on for the position in the genome zipper. So if you use the marker ID here, for example, and we search for it, you will get the corresponding position in the genome zipper where this marker was anchored. And if you click on a specific element, you can get more information about the specific gene. For example, here is a sorghum gene where you can get more information about that gene in the PLANT.db element report. And you can also go to the genome zipper report where you get a lot of information about a particular position in the genome zipper. For example, you get all the genes that were anchored from the reference organisms there. You get the chromosome, you get the centimorgan position, and you get all the elements that were tagged by that element in the genome zipper and anchored on that position. Now if you click this loci position on chromosome link here, you will get the corresponding sector on the chromosome, including the neighborhood. The yellow color indicates the current position on the chromosome and the centimorgan position. There is also a graphical representation. You are at the start of chromosome 1H and the neighborhood is given below and above. And you have the different sections, for example, full-length cDNAs that you can access. By clicking in, you can access the sequence of the full-length cDNA and more information. Again, you also have ESTs anchored there where you can get the sequence. And you can also download all the genome zipper data here in tabular format, for example in Excel format. And from the Barley PLANT.db instance, you can also access physical map data, where you can get more information about that and also access visualization tools just as gbrowse or crow's nest for the physical map. Okay, so the last part of the tutorial will be about how to download bulk flat files for the Barley genome data. Sometimes you want to work with your own copy of the data on your local machine, for example, to do some in-depth analysis on the data. And therefore we are running a download portal where we structured all the data. You can access it by clicking this download link here. And on the overview page we have the structure of the download portal. For example, we structured the data into anchoring, expression, genes, and sequences. And by clicking this FTP link, you are accessing the FTP server for the Barley download data. As you can see, there is a number of main categories such as anchoring, expression, genes, POPSEC data, sequences, or SNPs. And a README gives you some more information and an explanation of the different sections. There's also a data archive file which contains all data in one package. That's the Barley data archive here. And now we are going to have a look at the genes folder, where you will have a CiteMe and a README file. So we are going to have a look here. So there is a number of frequently asked questions. There is some explanation about the different files that are in that folder. And if you go back, you can see the different files which are separated into high confidence genes and low confidence genes. And next we are going to the sequences folder, where you can download the genome assembly. And the anchoring folder, it has a number of subfolders, which is a little bit more complex, so that's why we set up a README file, which gives you a lot of information about the files and the data included in that folder. And the SNP folder finally summarizes the variation data in Barley. So you now may have some questions about both the data that we introduced or the tools that we showed you. Please do not hesitate to contact us at these addresses here. We will be happy to help you. And last but not least, I want to thank and acknowledge all the partners that contributed data and helped. And thanks for watching.